Today, we're plunging into something truly fascinating and, well, foundational. It touches everyone's life, the incredible journey of drugs. We'll explore how they're discovered, how they actually work in your body, and, why they come in so many different forms. Our mission, to demystify this whole process for you.
It really is quite a journey. It's filled with some cutting-edge science, meticulous engineering, and details that, honestly, often surprise even people in the field. We're talking about pharmacodynamics. That's all about how drugs affect your body, and pharmaceutics, the, let's say, art and science of turning those drugs into usable, effective medicines. You're definitely going to gain a whole new appreciation for what goes into every pill or liquid or, cream you might encounter.
Okay, I'm ready. Let's unpack this. From what we've reviewed, it seems the life cycle of a drug starts, well, way before it ever reaches a pharmacy shelf.
Absolutely. You should think of it as a multi-stage marathon. It often spans, maybe 8 or 9 years before it even gets regulatory approval. It kicks off with the preclinical phase. This can last up to, say, 4 years.
4 years. Just the preclinical.
Yeah. This is all lab work, in vitro studies, using cell cultures, test tubes, and also animal testing. The goal here is to identify promising chemical compounds. Check if they actually work, how specific they are, and try to understand how they exert their effects. If a compound shows real promise, then an investigational new drug, or IND, application is filed. That's basically asking permission to start testing it in humans.
Okay, so if it looks good in the lab, then it takes that huge leap into human trials. And this is where it gets really interesting and incredibly rigorous, I imagine. What happens during this clinical testing phase?
This is definitely the longest and most critical phase. It can stretch anywhere from 4 to maybe 9 years.
Wow.
Yeah, and it's broken down into 3 distinct stages. Each one has its own specific purpose. Phase 1 involves a small group, usually 20 to 100 healthy volunteers.
Healthy volunteers, okay.
Right. Here, the focus is almost entirely on safety. And also pharmacokinetics, which means how the body handles the drug, how it's absorbed, distributed, metabolized, and, ultimately excreted. We're asking that fundamental question. Is this drug safe for people?
Makes sense. And if it passes that initial safety check, what's next?
Then we move to phase 2. This involves maybe 100 to 200 actual patients who have the condition the drug's designed to treat.
Ah, so now you're looking at efficacy.
Exactly. Now we're asking, does it actually work for this condition? And of course, we're still closely monitoring for safety. That never stops. If it keeps showing promise, it progresses to phase 3. This is a much larger trial involving maybe 1,000 to even 6,000 patients.
That's a huge number.
It is. This is the large-scale confirmation stage.
Okay.
We're rigorously testing efficacy again and extensively monitoring for any rare or unexpected adverse reactions, often using double-blind studies to make sure the results are unbiased. The question here becomes, does it work effectively in a broad population, and is it safe enough for general use?
Wow, that is just an incredible amount of effort and time and resources just to get one drug approved. It really highlights the rigorous standards. But even after phase 3, it's not immediately available, right? There's still another step.
That's correct. After successful phase 3 trials, a new drug application, an NDA, is submitted to regulatory bodies, like the FDA here in the U.S. If they approve it, then it moves to the marketing phase. And this includes what we call phase 4, or post-marketing surveillance.
Okay, so it's still being watched even after it's on the market.
Absolutely. This is continuous monitoring of the drug's safety and effectiveness in the entire general population. Because, new, perhaps rarer side effects might only show up when millions of people are actually using it. It's truly a lifelong watch, in a way.
Right. So we've explored this incredible journey a drug takes to get to you, but love to rewind just a little bit. What are we actually talking about when we say drug? What is that chemical at its core, and where do these powerful substances even come from?
Good question. At its heart, a drug is really any chemical substance that changes a physiological function and modifies a disease process. The World Health Organization, the WHO, gives us a pretty broad definition. Any substance used to modify physiological systems or pathological states for the benefit of the recipient. It's basically a tool, whether it's for healing, preventing illness, or even just exploring how our bodies work.
And they don't all come from some meticulously designed lab, do they? I gather some have surprisingly ancient natural origins.
You're absolutely right. We generally see 3 main types based on where they come from. First, you have natural drugs. These are extracted directly from plants, animals, microbes, even minerals.
Yeah.
Think of morphine from the opium poppy, or penicillin from fungi, or insulin, which originally came from animals. It's pretty amazing that some of our most potent medicines started right there in nature.
Yeah, it is. But I imagine isolating those compounds can be tricky. And maybe costly.
Exactly. While they can be powerful, they can be expensive and complex to purify. And there's always the potential for impurities. Then we have synthetic drugs. These are entirely created through chemical synthesis in a lab. Paracetamol, aspirin, those are perfect examples.
Ah, okay. Like common painkillers.
Right. These are often cheaper to mass produce, they tend to have high purity, and they're highly modifiable. Chemists can tweak their structure to get specific properties like making them more potent or reducing side effects. And finally, there are semi-synthetic drugs. These start as natural substances, but then they're chemically modified in the lab.
So a bit of both worlds.
Kind of. Amoxicillin, a common antibiotic, is a good example. Even heroin, which starts from morphine. This modification often allows for improved purity or stability or maybe activity compared to the original natural compound.
It's truly this complex ballet of actions happening inside us. But thinking about these categories, natural, synthetic, what's maybe a common misunderstanding people have about how drugs fix things, especially when you say they modify existing processes rather than introducing totally new ones?
That's a really great question. I think the biggest misconception is that drugs somehow introduce entirely new functions into your body. With the exception of maybe some very advanced gene-based therapies, that's generally not how it works. Drugs modify what's already there. They might turn up the volume on a process or turn it down or block a pathway completely or maybe replace a component that's missing. Think of them like skilled technicians tuning an existing machine, not installing a new one.
That's a great analogy.
And we see about 5 basic types of these modifications or actions. One is stimulation, boosting existing activity,
Yeah.
like adrenaline stimulating your heart activity during a crisis.
Right.
2 is depression or inhibition, reducing activity, like barbiturates depressing the central nervous system to help someone sleep. 3 is irritation. Now, this sounds negative, doesn't it?
Yeah, it does.
And usually, uncontrolled irritation is a harmful side effect. But sometimes, a controlled, localized irritation, maybe in certain topical creams, can provoke a desired response, like increasing blood flow to that area. It really highlights the fine line, between a therapeutic effect and an adverse reaction in drug design.
Interesting point.
4 is replacement. This is straightforward supplying something the body is lacking. Insulin for diabetes is the classic example, or levodopa for Parkinson's disease. And 5 is cytotoxic action. This means directly attacking harmful cells or organisms, like penicillin selectively targeting and killing bacterial cells without hopefully harming our own cells.
It's fascinating that they don't invent new processes, they just sort of tweak the existing ones. But how do they know what to tweak? What are these specific molecular targets that drugs aim for?
Ah, yes. That's the heart of pharmacodynamics, really. Drugs interact with specific large molecules or macromolecules in your body. We can generally categorize these targets into 4 main groups. First, enzymes. These are the biological catalysts that speed up chemical reactions in your body. Drugs can act as inhibitors.
Inhibitors, meaning they block them.
Right. They might block the enzyme's active site, kind of like putting the wrong key in a lock so the right key can't get in. Or they might bind elsewhere and change the enzyme's shape so it just can't function properly. Either way, this prevents the enzyme from forming its normal product. Many antibiotics, for instance, work by blocking essential bacterial enzymes, stopping the bacteria from growing or reproducing.
So it's about disrupting a natural process, often one that's vital for the target cell, like a bacterium.
Exactly. The second major target type is ion channels. Imagine tiny gateways in your cell membranes. These control the flow of ions, like sodium, potassium, calcium, in and out of cells. This flow is absolutely crucial for things like nerve impulses and muscle contractions. Drugs can interact with these channels in a few ways. They can be modulators, sort of gently fine-tuning how likely a channel is to open, or maybe how long it stays open, or they can be blockers, physically plugging the channel. Lidocaine, the local anesthetic, does this. It blocks sodium channels in nerve cells, which prevents the pain signal from traveling. That's how it causes numbness.
Wow. So drugs can essentially fine-tune or completely shut down these cellular gates. That's incredibly precise.
It truly is amazing. The third target group is transporter.
Transporters, like moving things around.
Exactly. These are specialized carrier proteins that move specific molecules across cell membranes. Drugs can inhibit these transporters, basically stopping them from doing their transport job. A great example is a class of antidepressants called SSRIs, selective serotonin reuptake inhibitors, like fluoxetine. They inhibit serotonin transporters in the brain.
Okay, so what does that do?
By blocking reuptake, they cause serotonin levels to increase in the synapse, the gap between nerve cells. This is thought to help alleviate symptoms of depression. It's a subtle but powerful way to influence brain chemistry. And the fourth, and probably the largest group of targets, are receptors.
Ah, receptors. I've heard that term a lot. The lock and key idea.
That's the classic analogy, yes. Receptors are regulatory macromolecules.
Right.
They're usually on the cell surface, but sometimes inside the cell. They recognize specific signal molecules, could be natural hormones, neurotransmitters, or could be drugs, and when that molecule binds, it initiates a specific response in the cell.
So different drugs can interact with these receptor locks in different ways then. It's not just one type of key.
Precisely. There are several key ways drugs interact with receptors. An agonist is like the perfect key. It fits the lock, turns it, activates the receptor, and produces an effect similar to the body's natural signal molecule. Morphine, for example, is an agonist at opioid receptors.
Okay, so it mimics the natural signal.
Right. Then you have an inverse agonist. This also binds to the receptor, but it actually reduces the receptor's baseline activity. It produces an effect that's the opposite of what an agonist would do. Some antihistamines actually work this way.
The opposite effect. Interesting.
Yeah. Then there's an antagonist. This binds to the receptor, fits in the lock, but it doesn't activate it. It just sits there, blocking the natural key or an agonist drug from binding and causing an effect. Naloxone, which is used to reverse opioid overdoses, is a powerful antagonist. It kicks the opioid off the receptor.
Wow, like a blocker.
Exactly. And then there's one more important type, a partial agonist. This one fits the lock and turns it, but only partially. It activates the receptor, but it never produces the full maximal effect, even if every single receptor is bound by the drug. Buprenorphine, used in opioid addiction treatment, is an example.
This is incredibly nuanced, and these receptors themselves, they aren't all simple switches, are they? I think our sources mentioned some really complex ones like G-protein coupled receptors.
You're absolutely right. G-protein coupled receptors, or GPCRs, are fascinatingly complex. Think of them as sophisticated cellular antennae, usually sitting on the cell surface. When an external signal like a hormone or a neurotransmitter binds to the outside part of the receptor, it's like a key turning a lock. But this doesn't just open a simple door.
Okay.
It triggers a whole chain reaction inside the cell. The receptor changes shape, activates an internal partner called a G protein, which then activates another enzyme. It's a cascade. It essentially amplifies that initial external signal to create a specific, significant cellular response.
So it's like a signal amplifier.
Exactly. Many of our body's most critical functions, vision, smell, immune responses, heart rate regulation, are controlled through GPCRs. They are hugely important drug targets.
And then there are receptors inside the cell. That seems counterintuitive. How does a signal from outside get to them?
Good point. Those are intracellular receptors. Only molecules that are lipid-soluble, meaning they can dissolve in fats, can easily diffuse right through the cell membrane to reach these receptors inside. Steroid hormones, like cortisol or testosterone, do this. And certain drugs are designed to do it too.
So once the drug gets inside and binds,
Once bound, the drug-receptor complex often travels to the cell's nucleus. There, it binds to specific regions of the DNA and essentially activates or deactivates specific genes. This leads to changes in the production of proteins, and it's these new proteins, or lack thereof, that carry out the drug's effects. It's generally a slower process than activating a surface receptor, but it can lead to very profound long-term changes in cell behavior.
so considering all this complexity, enzymes, channels, transporters, different kinds of receptors, internal versus external, what does this mean for designing new drugs? This structure-activity relationship idea must be absolutely critical.
Oh, absolutely. The structure-activity relationship, or SAR, is really foundational for drug design and development. It's the detailed study of how even tiny changes to a drug's chemical structure, adding or removing a functional group, changing its 3D shape, altering how fat-soluble it is, can dramatically affect its activity. How well does it bind? How potent is it? What side effects might it have? It's all about meticulously crafting the perfect chemical key for the specific biological lock you're targeting. Or sometimes designing a key that specifically gums up the works for an unwanted process, like in an invading bacterium.
Right, tailoring it precisely. And beyond these complex receptor interactions, our sources mentioned some drugs have even more direct ways of working, like pyrrolobenzodiazepines or PBDs. It said they directly bind to DNA.
Indeed, PBDs are a fantastic example of a non-receptor drug action. They literally function as DNA binding agents. They're shaped just right to snuggle into what's called the minor groove of the DNA double helix.
Wow, directly onto the DNA.
Yes. And some PBDs can even form crosslinks, like staples, between the two DNA strands. When they bind, They physically block the machinery that needs to access the DNA to read it and copy it, things like RNA, polymerase, and transcription factors.
Mm.
This effectively prevents gene transcription and protein synthesis.
Mm.
Ultimately, it leads to cell cycle arrest and triggers apoptosis-programmed cell death. It's a very direct attack on a cell's fundamental genetic machinery, which is why they are being explored as potent anti-cancer agents.
That's incredibly direct. Okay, before we move on from how drugs act, I want to circle back to something interesting the material brought up. Prodrugs. They sound really clever on paper, initially inactive compounds that only become active inside the body. So if they're such a neat idea, why don't we see them more often? Why aren't pharmacists pushing them more?
Prodrugs.
Yeah.
They are fascinating, it's true. The concept is usually to improve certain properties, maybe better absorption, better stability on the shelf, or perhaps reducing immediate side effects upon administration. Codeine converting to morphine in the liver is the classic example everyone knows.
Right.
But the reason they aren't the standard approach touches on some pretty significant challenges, both in how the body handles them, pharmacokinetics, and how they ultimately act, pharmacodynamics.
Okay, like what sort of challenges?
Well, probably the biggest one is unpredictable activation. The conversion from the inactive prodrug to the active drug usually relies on enzymes, often in the liver. But metabolic activity varies hugely between individuals.
Oh, so?
Genetics, age, liver function, even other drugs someone is taking, all these can affect how quickly or efficiently that conversion happens. Some people might be poor metabolizers for a specific enzyme.
Meaning they don't activate the drug well?
Exactly. So they might get reduced therapeutic effects or inconsistent effects. It makes dosing tricky. Then there's the complexity in formulation. You need to make sure the prodrug is stable before you take it, but then gets transported and converted properly after you take it. This adds cost and development hurdles.
I see.
Also, a delayed onset of action. Because it needs to be metabolized first, there's an inherent delay before the active drug starts working. That's often undesirable, especially in emergencies or for, say, acute pain relief.
Right. You want relief now.
Precisely. And regulatory and safety concerns. Regulators demand a lot of data on the metabolic pathways, proof of consistent, safe activation. Plus, there's always the worry that the intermediate metabolites the things formed during conversion could have unknown or even harmful effects themselves. And frankly, sometimes there are limited advantages. For many conditions, we already have effective conventional drugs. So the extra hurdles and costs for developing a prodrug aren't always justified if the benefit isn't substantial.
So from a practical standpoint, for a pharmacist...
Yeah, pharmacists often prefer directly active drugs because they're generally more predictable. It makes patient counseling, dosing adjustments, and monitoring for effects or side effects much more straightforward. Prodrugs just add another layer of complexity to manage.
That makes so much sense. It sounds like a really elegant idea in theory, but the real-world biological variability and practical complexities often make them less universally applicable than you might first think.
Okay, so taking all this incredible science, the targets, the actions, the prodrug complexities, how do scientists actually measure how safe a drug is? And what happens when different drugs interact inside the body?
Right, safety is paramount. This brings us to concepts like the therapeutic index, or TI, and the related idea of the therapeutic window. The TI is a crucial quantitative measure of a drug's safety. It's basically a ratio. The dose that causes toxicity in 50% of subjects, the TD50, divided by the dose that's effective in 50% of subjects, the ED50.
So toxic dose over effective dose.
Exactly. A small TI, like for the blood thinner warfarin, which is around 2, means there's a very narrow margin of safety. The toxic dose is really close to the therapeutic dose. These drugs require incredibly careful monitoring, often blood tests, to keep the patient in that safe zone.
Okay, narrow window, high risk. Pretty much. In contrast, a large TI, like for penicillin, which can be over 100, means it's much safer. The toxic dose is far, far higher than the dose needed for it to work. There's a lot more room for error. The therapeutic window is essentially a graphical way of looking at this. It shows the range of drug concentrations in the blood where it's likely to be effective without being toxic. It directly guides safe dosing strategies and helps compare drugs. It's a critical safety meter.
So this really guides safe dosing, patient comparisons, even drug development decisions. But what happens when you take more than one drug? Those interactions must be incredibly complex to predict.
They absolutely are, and understanding them is crucial for patient safety. Our sources highlight several main types of drug interactions. First, there's therapeutic antagonism. This is where one drug reduces or completely blocks the effect of another. Maybe they compete for the same receptor or have opposing physiological effects. Think of aspirin and ibuprofen, sometimes interfering with each other's antiplatelet effects if taken together. Or how acetylcholine slows the heart while noradrenaline speeds it up. They oppose each other.
Okay, one cancels out the other, at least partially.
Right. Then there's an additive effect. This is simpler. The effects of two drugs just sum up, usually because they have similar mechanisms or affect the same pathway. Think 1 plus 1 equals 2. For instance, combining certain NSAIDs, like ibuprofen, with glucocorticoids, like prednisone, can significantly increase the risk of gastrointestinal bleeding because both can irritate the stomach lining.
So, straightforward summation there, potentially leading to more risk.
Exactly. But then things get more interesting with synergism. Here, the combined effect is greater than the simple sum of the individual effects. Think 2 plus 2 equals 5 or maybe even 10.
Wow, really?
Yes, this can be incredibly beneficial therapeutically. A classic example is the antibiotic combination of sulfamethoxazole and trimethoprim. Individually, each might only be bacteriostatic, meaning they stop bacteria from multiplying. But together, they block sequential steps in a vital bacterial pathway. And the combination becomes bactericidal. It actively kills the bacteria. Much more powerful together.
That's a clever use of synergy.
Definitely. And finally, there's potentiation. This is where one drug, which might have little or no effect on its own in a particular context, significantly increases the effect of another drug.
So one drug boosts the other.
Precisely. For example, alcohol can significantly enhance the sedative or drowsy effects caused by some antihistamines, or it might even enhance aspirin's pain-relieving activity. This can sometimes be useful, but it can also dangerously increase the risk of toxicities.
This really underlines the incredible complexity of managing medications, doesn't it? What's the main takeaway for us, the listeners, about navigating these potential interactions?
I think the main takeaway, the bottom line, is how meticulously doctors and pharmacists must consider your entire drug regimen. Not just prescriptions, but everything. Even combinations that seem harmless on the surface can have significant, sometimes even dangerous impacts on how your body handles the drugs or how they work. This is exactly why it is so critically important to disclose all your medications to your healthcare provider. And that includes over-the-counter drugs, herbal supplements, vitamins, even things like strong teas or grapefruit juice, which can interfere with drug metabolism.
Right. It's not about being judged. It's about safety.
Absolutely. It's about connecting all the dots in a very complex biological system to keep you safe and ensure your treatments work as intended.
That's a powerful reminder. Okay, so far we've talked a lot about what a drug is and how it acts in the body. But how do we get that active chemical ingredient into a form that's safe, effective, and actually acceptable for you to take? This sounds like where pharmaceutics comes in.
Precisely. Pharmaceutics is that crucial bridge. It's the branch of pharmaceutical science that deals with the formulation, preparation, dispensing, and effective delivery of drugs. Its core objectives are, essentially, to take that new chemical entity, that raw drug, and convert it into a safe and effective dosage form, a pill, a liquid, an injection, whatever it might be. It's about ensuring the drug is stable on the shelf, that it's bioavailable, meaning it actually gets absorbed and reaches its target in your body effectively, and crucially, that it's acceptable and easy for you, the patient, to take correctly. Yeah. It's really where the raw drug science meets the patient's reality.
Okay, so let's start with maybe one of the most basic yet surprisingly versatile dosage forms, powders. What are their main advantages and, I guess, disadvantages?
Right, powders. They're simply finely divided drugs, sometimes mixed with other powders, in a dry state. Their big advantages include things like allowing for a wide range of ingredients and flexible dosing. You can measure out specific amounts, which is great for pediatric or geriatric patients who might need doses not available in standard tablets.
Oh, customizations.
Exactly. They often have enhanced stability and a longer shelf life compared to liquids because chemical degradation is often slower in a dry state. They can also offer rapid drug dispersion and absorption once they hit liquid in the stomach. And they can be cost-effective, especially for compounding specific prescriptions.
Okay, sounds pretty good. What are the downsides?
Well, the obvious one is often unpleasant taste, which can be hard to mask in a powder. There's also potential for dosing inaccuracy if the patient isn't careful with measuring, especially with bulk powders. They can be bulky to carry around. There's a risk of spillage. And they're not suitable for drugs that are unstable when exposed to air or moisture, or may be degraded by stomach acid if taken orally. They can also absorb moisture from the air, which can cause stability issues.
Okay, so a mix of pros and cons, like anything. But let's look at liquid medications. They're incredibly common, especially for kids or people who have trouble swallowing pills. What makes them different?
Liquid dosage forms are definitely popular. They involve the active pharmaceutical ingredient, API, and various inactive ingredients or excipients, either dissolved, suspended, or dispersed in some kind of liquid vehicle, usually water-based, but sometimes oily. The main appeal, as you said, is ease of swallowing, and often they offer rapid and potentially more uniform absorption compared to a solid form that first needs to dissolve. We broadly categorize them into two main types: monophasic liquids.
Monophasic, meaning one phase.
Exactly. These are homogeneous, clear solutions where all the components are completely dissolved in a single liquid phase. Think of syrups, elixirs, tinctures, even mouthwashes. They generally offer the most rapid and predictable absorption. Then there are biphasic liquids, bi meaning two. These contain two distinct phases that don't fully mix or dissolve into each other.
Okay, two phases, like oil and water.
Precisely. The two main types of biphasic liquids are suspensions and emulsions. Suspensions have fine, solid particles dispersed in a liquid. Emulsions have tiny droplets of one liquid dispersed throughout another liquid that it doesn't mix with, like oil droplets in water.
Right. And our sources gave us a lot of details specifically on emulsions and suspensions. Let's start with emulsions. What exactly are they? And I guess, how do pharmacists even tell what kind they're dealing with?
Okay, an emulsion is technically a biphasic liquid dosage form where one liquid phase, the dispersed phase, is distributed as tiny droplets throughout another immiscible liquid phase, the continuous phase. Think of milk again. It's tiny fat oil droplets dispersed in water. They are by nature thermodynamically unstable. Oil and water just don't want to stay mixed, so they always require emulsifying agents, special ingredients to stabilize them and prevent the droplets from merging and separating out.
So keeping the oil and water mixed. And you mentioned types.
Yes, we primarily classify them based on which liquid is the dispersed phase and which is the continuous phase. You have oil and water, OW emulsions, where tiny oil droplets are dispersed in a continuous water phase. These are generally preferred for oral use or injectables because they mix readily with body fluids. And you have water and oil, WO emulsions, where tiny water droplets are dispersed in a continuous oil phase. These are often used for external applications, like creams and ointments, because the oily continuous phase can provide moisturizing or protective properties on the skin.
Okay, OW versus WO. How do you actually identify which is which in the lab? Can you just look at it?
Not always reliably just by looking. There are several standard identification tests, and they're actually quite clever. The dilution test is simple. You try to mix a drop of the emulsion with water. If it mixes easily and uniformly, the continuous phase must be water, so it's an OW emulsion. If it doesn't mix, it's likely WO.
Makes sense.
Then there's the conductivity test. Water conducts electricity much better than oil. So if you put electrodes in the emulsion, an OW emulsion will conduct electricity well because water is the continuous pathway. A WO emulsion will show very little conductivity.
Ah, using electrical properties. Neat.
Yeah. There are others too, like using dyes that selectively dissolve in either water or oil, or looking at fluorescence under UV light, as oils often fluoresce.
That's really fascinating. So these emulsifying agents you mentioned are clearly key to making emulsions work at all. How do they actually perform that magic of keeping oil and water mixed?
Emulsifying agents, which are often surfactants, are really the heroes here. They are special molecules that have both a water-attracting hydrophilic part and an oil-attracting lipophilic part. They work primarily by reducing the interfacial tension, the natural repulsion between the oil and water surfaces. They position themselves at the interface between the oil droplets and the water, forming a protective film or barrier around each tiny droplet.
Like a little jacket for each droplet.
Sort of, yes. This film prevents the droplets from bumping into each other and merging or coalescing. Some emulsifiers also increase the viscosity, the thickness, of the continuous phase, which physically slows down the movement and separation of the droplets. It's all about creating and maintaining that stable dispersion.
And our sources mentioned something called an HLB value. What's that about?
The HLB value. That stands for hydrophile-lipophile balance. It's a numerical scale, typically from 0 to 20, that quantifies how much an emulsifier likes water versus how much it likes oil. A low HLB value, say 0 to 6, means the emulsifier is more lipophilic or oil-loving. These are generally good for making W emulsions. A high HLB value, say 8 to 18, means it's more hydrophilic or water-loving. These are preferred for making OW emulsions.
So it helps you pick the right tool for the job.
Exactly. It helps formulators select the most appropriate emulsifying agent, or often a blend of emulsifiers, to achieve a stable emulsion of the desired type, OW or WO. For example, the sources noted a blend giving an HLB of 10.72 would be suitable for an OW emulsion. It's a very practical tool.
And what are the classic ways pharmacists actually prepare an emulsion? I think I read something about a crackling sound being involved sometimes.
Ah, yes, the preparation methods. There are a few standard techniques, often done using a mortar and pestle. One very common one is the dry gum method, also called the continental method or the 4.2.1 method, because of the typical ratio of oil to water to gum emulsifier.
Dry gum, so you start dry.
Yes, you mix the gum emulsifier, like acacia, thoroughly with the oil first, in a dry mortar. Then you add all the required water at once and triturate, grind, and mix vigorously immediately. As the primary emulsion forms rapidly, you often hear a characteristic crackling sound. It's quite distinct.
Huh, a sound indicator. What's the other main way?
The other main one is the wet gum method, or English method, which also often uses a 4.2.1 ratio. Here, you start by making a mucilage. You mix the gum emulsifier with the water first to form a thick, sticky liquid. Then you add the oil gradually in small portions, while triturating continuously after each addition. This method is often slower, but can sometimes yield more stable emulsions.
Okay, dry gum versus wet gum. Got it. But even with all that careful preparation and the right agents, emulsions can still go wrong, right? They can break down.
They absolutely can. As we said, emulsions are inherently unstable systems just waiting for a chance to separate. Several types of instability can occur. Creaming, or sedimentation, is when the dispersed droplets either rise to the top, creaming if they're less dense, like oil and water, or sink to the bottom, sedimentation if they're denser. This is usually reversible just by shaking the bottle.
Okay, so shaking fixes that. What's worse?
Flocculation. This is when the droplets clump together to form loose aggregates, or flocks. They haven't merged yet, but they're sticking together. This can sometimes be reversed by shaking, but it's a step closer to breaking. Then you have coalescence. This is where the droplets actually merge together to form larger and larger droplets. This process is irreversible. Once they merge, you can't get those tiny droplets back just by shaking. This also leads to, exactly, phase separation, also called breaking or cracking. This is the complete irreversible separation of the emulsion back into its distinct separate layers of oil and water. The emulsion is destroyed.
Right, you definitely don't want that.
Yeah.
Okay, what about the other type of biphasic liquids, suspensions? What are their unique challenges and the solutions formulators use?
Right, suspensions. These are preparations containing finely divided, insoluble solid drug particles distributed somewhat uniformly throughout a liquid medium in which the drug is not readily soluble. They're very useful for administering drugs that are poorly soluble in water, which is actually quite common. They can sometimes offer higher bioavailability than tablets for certain drugs, allow flexible dosing, and can be good for masking the unpleasant taste of a drug particle inside the liquid.
So good for insoluble drugs. What are the challenges?
The main challenge, similar to emulsions but different in detail, is physical instability. The solid particles will inevitably tend to settle down over time due to gravity. This is called sedimentation. The real problem arises if these settled particles pack together tightly at the bottom to form a hard, dense sediment called a cake. Caking is often irreversible. You can shake the bottle all you want, but that hard cake won't redisperse properly.
Ah, so caking is the big enemy for suspensions.
It really is. If the patient can't redisperse the sediment uniformly, they won't get the correct dose. So a good suspension is designed so the particles don't settle too readily, but if they do settle, the sediment formed should be loose and easily redispersed with minimal shaking without forming a hard cake.
Okay, so how do formulators actually achieve that? How do they prevent caking?
This is where the fascinating concept of flocculated versus deflocculated suspensions comes in. It's a bit counterintuitive. In a flocculated suspension, we actually encourage the particles to form loose, lightweight aggregates called flocks. These flocks settle faster than individual particles would.
Faster settling sounds bad.
You'd think so, but the key is that they form a loose, fluffy, porous sediment network at the bottom. This sediment is very easy to redisperse with just gentle shaking. The liquid above the sediment, the supernatant, tends to become clear quickly because the particles are all in the flocks. This is generally the preferred state for oral suspensions to prevent caking and ensure dose uniformity after shaking.
Okay, so faster settling but easy redispersal. What's the alternative?
The alternative is a deflocculated suspension. Here, the individual particles remain separate and discrete, often because they have repulsive forces between them, like a high electrical charge or zeta potential. These individual particles settle much more slowly.
Slower settling sounds good.
It seems good initially. The suspension stays cloudy longer. But the problem is, when they do eventually settle over time, These individual particles can pack together very tightly, layer by layer, forming that hard, dense cake we talked about, which is extremely difficult or impossible to redisperse.
Ah, so slow settling leads to hard caking. I see the trade-off now. Flocculated is usually better for oral use.
Generally, yes. Deflocculated systems might be preferred only in specific cases, like maybe some parenteral, injectable, or ophthalmic eye drop suspensions, where very slow settling and a uniform appearance for longer periods might be critical, and caking might be less likely under those specific conditions or formulations.
So what kinds of additives do formulators use to control this behavior to encourage flocculation or slow settling or keep things dispersed?
They use a whole toolkit of excipients, each with a specific role. Flocculating agents are used specifically to promote the formation of those loose flocks. These can be electrolytes, surfactants, or certain polymers that reduce the repulsive forces between particles. Suspending agents, or viscosity modifiers, are crucial. They increase the viscosity or thickness of the liquid medium. This physically slows down the rate at which particles or flocks settle. Think of things like xanthan gum, methylcellulose, or tragacanth.
Like making the liquid thicker so things sink slower?
Exactly. Wetting agents are important too, especially for drug particles that are hydrophobic, water-hating. These agents help the liquid vehicle to displace air from the surface of the particles and coat them properly, allowing them to be dispersed in the water-based liquid. Polysorbates are common examples. You might also have diffusing agents to help maintain uniform particle distribution, preservatives to prevent microbial growth, since many suspensions are water-based, and of course, organoleptic additives, sweeteners, flavors, colors to improve taste, smell, and appearance, especially for oral liquids.
Wow, quite a balancing act to get a suspension just right. Okay, finally, let's talk about tablets. Arguably the most common drug form we encounter. How are they typically made, and what ensures their quality?
Right, tablets. They are solid dosage forms, usually made by compressing a powder formulation. There are three main manufacturing methods, and the choice depends on the properties of the drug and other ingredients. The simplest is direct compression. Here, you just blend the drug with suitable excipients that have good flow and compressibility properties, and then directly compress the mixture into tablets. It's fast, cost-effective, and good for drugs sensitive to heat or moisture. But it really requires the powder blend to flow exceptionally well.
Okay, simple if the materials cooperate. What if they don't?
Then you often turn to granulation methods first, to improve the powder properties before compression. Wet granulation is very common. You mix the drug in fillers, add a liquid binder solution to form a wet mass, pass it through a screen to form wet granules, dry the granules, screen them again for uniform size, add lubricant, and then compress them into tablets.
That sounds much more involved.
It is. It improves flow and compressibility, reduces dust, prevents segregation of ingredients, and is good for low-dose drugs. But it's complex, time-consuming, involves multiple steps, and isn't suitable for drugs degraded by moisture or heat used in drying. The third method is dry granulation. Here, you compact the powder mixture without using liquid, either by forming large slugs in a heavy-duty tablet press, slugging, or by pressing the powder between rollers, roller compaction. These compacted masses are then milled into granules, blended with lubricant, and compressed into the final tablets.
So no liquid involved. Good for sensitive drugs.
Exactly. It's great for moisture or heat-sensitive drugs and improves flow and compressibility, but it often requires specialized, heavy equipment.
And you mentioned excipients again. These sound like the unsung heroes of tablets doing all the work besides the actual drug.
They truly are. Excipients are the inactive ingredients, but they are absolutely crucial for making a functional, stable, effective, and acceptable tablet. They perform so many vital roles. You have diluents, or fillers, like lactose or microcrystalline cellulose, mainly to increase the bulk of the tablet to a practical size for handling and compression, especially when the drug dose is very small. Binders, like starch paste or povidone, PVP, are added to promote the adhesion of powder particles together, forming the granules needed for good compression and tablet hardness. Disintegrants, like cross-carmellose sodium or cornstarch, are critical. They are added to help the tablet break apart quickly into smaller fragments when it comes into contact with fluids in the gastrointestinal tract, which allows the drug to dissolve and be absorbed.
Ah, so the tablet actually needs to fall apart efficiently.
Yes, absolutely. Then you have lubricants, like magnesium stearate. These are added just before compression to reduce friction between the tablet surface and the die wall during ejection from the tablet press, preventing sticking. Gleadins, like colloidal silicon dioxide, are added to improve the flow properties of the powder blend, ensuring the dyes fill uniformly during compression. You can also have release modifiers to control how the drug is released, for example, for extended release tablets, coating agents for protection, taste masking or controlling release location, and of course, sweeteners, colorants, and flavorings for patient acceptance.
It's amazing how many different components go into making even a simple-looking tablet work correctly. So once a batch of tablets is made, how do manufacturers know they're good quality? What tests do they run?
Tablet evaluation is essential, a critical part of quality control. Several key tests are performed routinely. General appearance. Just a visual inspection for any defects like cracks, chips, discoloration, or sticking. Weight variation. They weigh a number of individual tablets to ensure they're consistent. Uniform weight generally implies uniform drug content if the blend is homogeneous. There are official limits, for example. The USP might allow only 5% variation for larger tablets. Content uniformity. This is even more specific. They actually assay individual tablets to measure the amount of active drug present, ensuring it's within a tight range, often 85% to 115% of the labeled amount. This is critical for ensuring correct potency in dosing.
Okay, consistency is key. What else?
Hardness or crushing strength. They measure the force required to break a tablet. It needs to be hard enough to withstand handling and shipping without breaking, but not so hard that it fails to disintegrate properly in the body. A typical range might be 4 to 8 kJ meeting position. Friability. This tests the tablet's resistance to chipping and crumbling during handling, like tumbling in a special rotating drum. Usually, less than 1% weight loss is considered acceptable. And perhaps most importantly for efficacy, dissolution. This test measures how quickly the active drug dissolves from the tablet into a specific liquid medium under controlled conditions. This is vital because the drug generally needs to dissolve before it can be absorbed into the bloodstream. There are specific requirements for how much should dissolve within a certain time.
This deep dive has truly taken us on a remarkable journey, hasn't it? From the intricate molecular dance of pharmacodynamics, how drugs work at the deepest level, to the careful practical engineering of pharmaceutics, how we actually make medicines you can use. Every time you encounter a medicine now, hopefully you'll have a much deeper understanding, a real appreciation for the incredible science and effort behind it.
Indeed. We've really only scratched the surface today, but even just understanding the difference between, say, an agonist and an antagonist, or appreciating the challenges in making a stable emulsion or a tablet that dissolves correctly, it gives you a powerful new lens through which to view the world of health and medicine. It's truly a testament to decades of human ingenuity and rigorous scientific inquiry, all aimed at improving health.
So, what does this all mean for you listening right now? Well, next time you reach for any medication, whether it's a pill, a liquid, a cream, maybe take a moment to consider the remarkable journey it took to get to your hand. Think about the precise molecular mechanisms likely at play inside your body, the meticulous design of its formulation, and the incredible effort put into ensuring it's not just effective, but also stable, reliable, safe, and easy for you to take. It really is a marvel of science and engineering working hand in hand for your well-being, often hidden in plain sight in your medicine cabinet.